A very good morning, students. In the last class, we were discussing about the immunity and we studied about the innate immunity, what is innate immunity and what are all its different types of innate immunities. And in today's class, we are going to discuss about the acquired immunity and their components. Now, we'll just come back to once again what is this acquired immunity. The acquired immunity a person will get after the birth. So it is after birth. If a person is affected with any kind of a disease in his lifetime, he acquired certain immunity and that immunity is called acquired immunity. And we also discussed about the unique features of this acquired immunity, that is the antigenic specificity, diversity, recognizing self and non-self antigen and memory. So these are all the unique features of this acquired immunity. And today we are going to discuss about the components of this acquired immunity. Now before that, we'll just discuss about two important topics in this acquired immunity. One is the active immunity as well as passive immunity. Active and passive. So these are all the two things that we will discuss now. What is this active and passive? These are all the types of this acquired immunity. So first we will discuss the type and then we will discuss about the component. Now the active immunity, in the sense, here the antigen is having a contact with the immune system. After the antigen or a person is affected with the disease, so the antigen will cause a kind of infection, okay, as well as this because of this infection or because of this antigen, the infection is caused and in the person's body antibodies are produced against this antigen. That is what we discussed yesterday. So these kind of antibody, after the antigen is encountered and due to the infection, the antibodies are liberated. This kind of immunity is called active immunity. That is, it is produced actively by host immune response. So active immunity is generated due to infection, number one. It is also generated due to immunization method. Immunization means taking vaccines. So vaccine is a attenuated antigen. Vaccines we will study later on in detail. But now what we are doing about what we have to know about vaccine is there are an antigen which is going to cause disease. But in vaccine what we are doing is we are killing that antigen, we are removing the toxic and inserting only the antigen inside. So the antigen enters inside the body, it cannot infect. Why? Because there is no toxic substance. We remove the toxic. So only antigen is inserted inside. And for that antibodies are synthesized. So antibodies are produced after an infection or after an immunization. And this antibody is called active immunity. Whereas in passive immunity, there is no involvement of antigen. That means antigen is not enter into the body. Okay. So that means how antibodies are produced? Antibodies are not produced in the person's body, so that antibodies are given from outside. So somewhere we are preparing the antibody or for some other reason. So even this passive immunity, the mother's milk is a very good example for the passive antibody. So mother's milk will have more antibodies. When the mother feeds the fetus, the antibody will be inserted or taken into the fetus body. So that kind of an antibodies are called passive antibody. Whereas active antibody will activate it only after the antigen has encountered. So one more thing that you have to understand in this, whenever antibodies are produced, what happens is along with the antibody, some memory cells also will be produced. Why? because there are certain cells, a particular cell we will talk after this class, that cell will produce this antibody when the infection is there. At the same time, some memory cells also will be produced, and this memory cell only will involve in secondary immune response. Second time when the same antigen is encountering the body, the antibody production will be done faster. Okay, so this active immunity, there are memory cells are involved, but there is no antibody production in the body. So the antibody is given from outside. So here there is no memory cells involved. There is no memory cells. Understand? Right. So the antibody which is here it is produced will always be effective and it is durable, but whereas it is less effective. Understand? So that is what very important thing regarding this active and passive. So for this you can just refer the book also, but these 2 points are very important regarding this types of acquired immunity. Is that clear? Now we will talk about the components of acquired immunity. Right. So what are all those components? So we are talking about acquired immunity and we are going to discuss about this components. See, components means with what the acquired immunity is being developed. So when we talked about this innate immunity in yesterday's class, we know the innate immunity was performed by certain barriers like anatomical barriers, physiological barriers, phagocytic barriers, and certain chemicals and interferons and all this. Now here this acquired immunity has been done by certain components. So there are 2 components are there. One is called CMI and other one is called AMI. First I will tell you what is the CMI and AMI. Then we will discuss in detail about the CMI and AMI. CMI, the expansion for CMI is called cell mediated immunity. Okay, so here what is happening is the cell is going to involve in destroying the pathogen, the antigen, whereas AMI is otherwise called as antibody mediated immunity. It is also called as humoral. Humoral means humoral. The humoral mediated immunity means the antibodies are produced will always be or situated in the plasma of the blood. So it is a fluid part. Okay, so plasma is a fluid part. Only the RBC is a solid part that we know. So where these antibodies, the antibodies are there in the fluid called plasma. Even if you take your blood and check, you will have some antibodies. So that is there in the plasma. So that is why it is called humoral immunity. Antibody mediated immunity is also called as humoral immunity. Understand, right? So now here there are 2 types of acquired immunities are there. One is cell mediated. Cell means with the help of cell an antigen is being destroyed. But whereas in antibody mediated immunity, with the help of antibodies the antigen is destroyed. So our aim is to destroy this antigen. So we can destroy by using the cell, or you can destroy by using the antibodies. So depend upon the role played by this immunity, they are called as cell mediated immunity or antibody mediated immunity. Whether if it is attacking by the cell, it becomes cell mediated, or with the antibody it is called antibody mediated immunity. Understand? So first we will talk about what is the cell mediated immunity, and then we will go for this antibody mediated immunity. First we will talk about the basic thing. Now a pathogen is destroyed by cells. I told you an antigen, this antigen will be destroyed by cells. That means there is no antibody production. In cell mediated immunity, always remember there is no antibody production. Okay, so then that means how it is being done. Now the cell mediated immunity is done by certain cells in the body. Those cells are very important, T cells. I will talk about these T cells. T cells. Second one, it is NK cells. NK means natural killer cells. And the third one is the macrophages. Macrophage is one type of WBC. Out of 5 types, monocyte is the WBC. Whenever a cell is getting infected, monocytes will be converted to macrophage. Don't forget it. Yesterday itself we have discussed about this. So cell mediated immunity is done with help of cells. There is no antibody, and those cells are T cells, natural killer cells, and macrophages. That much clear, right? So now antibody mediated immunity. In antibody mediated immunity, the antigens are destroyed with the help of antibodies. Okay, and this is done with the help of cells called B cells. So remember, cell mediated immunity is done by T cells and antibody mediated immunity is done by B cells. All these are 1 mark questions. Okay, B cells. Then apart from B cells, it is also done with the help of T helper cells, not T cell, T helper cell. And one more cell is there called APC. APC means antigen presenting cells. So these are all APCs for antigen presenting cells. So these are the cells is involving for the elimination or the destruction of the antigen. Okay, so we have cell mediated immunity as well as we have got the antibody mediated immunity. Now how this is being working out, how the antigen is being destroyed, that we will see now, and then we will complete this with a flow chart. Okay, so this antibody mediated immunity and cell mediated immunity, now we will, now little extra we will discuss. Okay, so now here there are, first we will talk about the cell mediated immunity. So we are going to talk about the cell called T cells. T cells. Now T cell, actually the T cells are produced in the bone marrow. It is produced in bone marrow. Bone marrow, it is present inside the bone. So now one more thing that you understand, this kind of antibody mediated immunity is present only for the vertebrates. Those who have backbone will have this. The other organisms do not have this. Now the cell mediated immunity, here it is done by T cells, and the T cells have bone marrow. They are produced in the bone marrow. But the T cell cannot get matured in the bone marrow. So production is in the bone marrow and maturation in the organ called thymus. Okay, thymus. So that is why, since the T cell are getting matured in the thymus, it is having the name called T for thymus. Okay, so the production is in the bone marrow and the maturation in the thymus. That is what about the T cells. Now, you know the role of this antigen now. So what the antigen does? The antigen will enter into the human body and it will cause infection. This is what the role of this antigen. Okay, right. So I will just erase this entire thing, and as a flow chart I will explain to you. So we are talking about the cell mediated immunities, that is called the CMI. CMI separately we will first talk CMI. In CMI we are talking about the cell called T cells. We know where it is produced, we know where it is matured, right? So now we are talking about the antigen. This is the antigen. What is the role of antigen? Antigen involves in infecting our body. So it causes infection. Okay. Now, if it is causing infection, ultimately it is going to produce immune response, that is antibody production will be done. Okay. So now these antigens are recognized by our immune system. How this antigen, now this antigen is producing this antibody. Now what this antibody will do? This antibody will go and bind with this antigen and destroy. But this antibody is produced from the antigen only. But still the antibody will destroy this antigen. Now how this antibody will understand, will recognize this antigen is, every antigen has a protein on the surface like this. So this diagram you may be very familiar. in all the advertisement, all the corona, everything, you can find this diagram. So this protein is called GP. GP is called as glycoprotein. So this antibody, or we will call it as immune system, then only we can come to this antibody, okay, because first immune system has to recognize it. So when the antigen infect the human body, the immune system has to recognize this antigen. How the immune system recognize the antigen? One, it will get activated first as soon as it enters. Then it will go and bind with the receptors. So this is a glycoprotein. So this glycoprotein will act as a receptors. It is called as receptors. Okay. So the glycoprotein will act as a receptors. Okay. Right. So now this glycoprotein, which is presented on the surface of this antigen, will act as receptors for certain cells that is present in the immune system. Okay. Immune system will not go immediately or straight away and bind with this receptor. Immune system will have certain cells. That cells only we talked, that is the T cells, then B cells, then we have got some macrophages, okay, then macrophages, then we have got some dendritic cells and some neutrophils, then we got some cytokinin. So all these are the cells which is present inside the immune system. Clear? Now we are getting, no? Once the antigen enters into the body, it causes infection. At the time, your body's immune system is activated, and this immune system has to recognize the antigen. How? It will recognize the receptors. So who will go and recognize it? The cells which is present in the immune system will recognize that. Okay. So that means this is also having receptor. So these cells also will have receptors. So suppose if this is a T cell and this T cell and this is the B cell, and these cells also will have receptors like this on the surface. So naturally this will go and bind with this. If this is a B cell, it will go and bind with this like this. So this is called receptors. Okay, so all these cells will have receptors to receive this antigen in order to destroy this. Understood? So all these are having glycoprotein in the surface. This much clear? Right. So now every cell in this immune system has a unique feature in defense. Everything individually, it has the capability of fighting with the antigen. Clear? So first we will talk about the macrophages. Why macrophages? The first cell to encounter the antigen. If an enemy enters into the body, the first person to go and fight with that enemy is the macrophage. Clear? So that is the reason we are studying the macrophages now. So what the macrophages will do, where the macrophages are? The macrophages are in the spleen and lymphoid organ that we will study. Right. So what this macrophages will do? The macrophages will do a process called phagocytosis. That means if this is an antigen and this is a macrophages, and the macrophages will form a pseudopodia and it will take this antigen inside and detoxify the antigen, and after detoxifying this antigen, it will present the antigen on the outer surface. It will break the antigen and present the antigen on the outer surface like this. Okay. So if this is a receptor, in the receptor antigens are presented like this. So all these having receptors. The receptor antigens are present. So macrophages will capture that antigen, process the antigen and present on the surface by the process called phagocytosis. Is that clear? Right. So after presenting this, now the macrophage will give signal to the T cells. So after this, the macrophage will give signal to the T cells. Okay, we are talking about cell mediated immunity, that is what T cells. Now there are two type of T cells are there. One is called T helper cell, one is called T helper cells. Okay, the another cell is called T cytotoxic cells. So there are two cells. It is also called as NK cells, natural killer cells. So there are two T cells are there, T helper and T cytotoxic cells. Now what this macrophage will do, after doing phagocytosis, it will give signal to the T cells. By the time these two T cells will go and bind with this presented antigen, and then they get activated. Okay, this much you are clear, right? So now I am talking about this T cytotoxic cell first, then we will see the T helper cells. Now when you talk about CMI, you should talk about the T cytotoxic lymphocyte. Why T cytotoxic cells is, it will create some pores. This cell is affected with virus, no, macrophage will has engulfed this and this is being presented. Now it will create some pores on the surface of the body. That pore is called Perforin Pores. Okay, the natural killer cells are T cytotoxic. Cyto means cell, toxic, making cell toxic, poison. So it will create a pore on it, and that pore is called perforin lined pores. Inside the pore, it is allowing water to enter inside. The water enters inside and the cell swells. When the cell is swelling, it will burst and the entire cell is ruptured. The entire infected cell is ruptured. That is what cell mediated immunity is. There is no antibody involved. So far we have not studied about antibodies. So cell mediated immunity with the help of cells. Okay, this infected cell is destroyed. Understood? That is why it is performed by macrophages. Then it is performed by T cells and cytotoxic T cells, or otherwise called as NK cell. You remember, cell mediated immunity is performed by three cells: macrophages, T cells and NK cells. Is that clear? Right. Second one is AMI. So from up to this we have studied CMI. Now we are going for antibody mediated immunity. Clear? Right. So in antibody mediated immunity, it is now two cells are get activated by the macrophages. One is called T helper cell and T cytotoxic cells. Okay. Now this activated T helper cells. Activated T helper cells. Now what this will do? How it is activated? By the macro. Who is activating? Macrophages. Now this T helper cell, now when it is getting activated with the help of macrophages, it will send signal to the B cells, or otherwise called as B lymphocytes, because in WBC out of 5 types, one type among that is called lymphocyte. What are the 5 types? Neutrophil, eosinophil, basophil, lymphocyte and monocyte. So lymphocytes. Lymphocytes are 2 types, B cells and T cells. T cells we studied. Now what about B cells? B cells produced in the bone marrow get matured in the bone marrow, but T cells produced in the bone marrow matured in the thymus, whereas B cell produced in the bone marrow matured in the bone marrow, that is why it gets a name called B cells. Okay, so once the T helper cell is activated, it activates B cells. Once the B lymphocyte activate, it is produced in the bone marrow, all this, it produces 2 type of cells. Okay, that first cell is called the plasma cells. Plasma cell. And plasma cell produces antibodies, and this antibody will recognize the antigen and it will destroy the antigen. Okay, so this plasma cell produces antibodies, and this antibody recognize the antigen and destroy the antigen. So that is the role of first activation of the B cells. So where this antibody, plasma cell, that is why this is called AMI, antibody mediated immunity. And this activates 2 things I told you. One is plasma, another one, most some only produce antibody. Some B cells activated produce plasma cell. Some B cells produces memory cells, memory B cells for doing secondary immune response. Clear? So who produces antibody? Not plasma. B cell produces antibody. Very important 1 mark question. B cell produces antibody. How it is produced? With the help of T helper cells. So antibody mediated immunity is performed by, already we studied, that is called the B cells as well as T helper cell, and this is activated by macrophages here. So that is called antigen presenting cells. Macrophages presenting the antigen on the surface. So it is called antigen presenting cells. Clear? So that is what the entire immunity, that is acquired immunity. So yesterday we discussed innate immunity, and today we have discussed about the innate as well as the acquired immunity. So far we have completed. I hope you understood what is this complete picture of this acquired immunity. So in this cell mediated, antibody mediated immunities are very important. You have to remember the sequence in order. If you can do this order, you can remember, you will never forget. It's a beautiful concept. You have to understand step by step so that it will be interesting for you to learn more and more. Okay, this is only a part of that given in your syllabus. When you come for the higher classes, you will study more regarding the CMI and AMI. Clear? Right. Topic I will add along with this, because so far we were discussing about this B cell and T cell and all. So one more topic also along with this you can study, that is cells of immune system. What are all the cells which is involved in the immune system? That is the cells of immune system. Right. So along with this we will just complete it. Now, there are so many cells are there in the immune system. The very important one is all the cells are interdependent cells. They do not depend upon the other cells, but still they protect the body from certain microbial infections as well as some growth of the tumor cells. Excuse me. Now, the bone marrow is the main one, which consists of the potentiality of producing all the cells in our body. Okay, the diagrammatic pictures will be given in your book. You can refer afterwards. I'll just give you an easy way of understanding the entire cells which is occupied in the immune systems. Already we have studied, but still. Now, in this, the bone marrow has the potentiality of producing all the cells in the immune system. So bone marrow consists of pluripotent. Potent means potentiality. Pluri means it has the potentiality of giving rise to various cell types. So bone marrow cells are the stem cells. So what kind of a stem cell it is? Stem cell or here pluripotent hematopoietic stem cells. Why hematopoietic stem cells? Hematopoietic means blood cells are formed. So here pluripotent hematopoietic stem cells means in the blood not only RBC is there, not only plasma is there, we got so many other cells like WBCs, 5 types of cells are located inside. So all these cells are produced from a single bone marrow. That is why in bone marrow we call it as pluripotent hematopoietic stem cells. Is that clear? Right. Now, this pluripotent hematopoietic stem cells it generates 2 types of cells. Number 1 is called the myeloid progenitor. Progenitor means before, it is a mother cell, from this other cells are being formed. So one cell is called myeloid cells, another one is called the lymphoid progenitor cells. Lymphoid progenitor. So that means from this the other lymphoid cells are produced. First we will talk about the lymphoid cells and then we will study about the other cells. Now, when you talk about the lymphoid, because the majority of the cells which is involving in immune function is the lymphocytes. In that lymphocyte around, because it is capable of recognizing and it will produce some immune responses. Okay, so immune responses are majoritarily produced from the lymphocytes. So there are around 20 to 30% in the WBC, 20 to 30% we have this lymphocytes. And you can find it is having a large nucleus. So if this is the lymphocyte, it is having a large nucleus inside. Okay, and it is having surrounded by some cytoplasm. So this is a nucleus and this is a cytoplasm. Okay, now this cells only we studied already, that is the B cells as well as the T cells. Clear? So these are the two lymphocytes. Don't forget it. These are the two lymphocytes. So if you can study CMI, AMI, we need this B cells and T cells. So this is the lymphocytes. Okay. And this lymphocyte has the receptors and all, that is B cell receptor. B cell will produce antibody that we already studied. And this is having the T cells will have two cells, that is the T helper cell as well as natural killer cells. Okay, and this all this will have receptors, so it is called B cell receptors and T cell receptors in order to bind with the antigen and destroy all this, making it to destroy. Okay, so these are all the cells, and this B cell, whenever it is produced, it is also produce some plasma cell also, plasma cells. So B cells, antibody, plasma cells, and memory cells are produced. So this is from that is the main object of this lymphocytes. So lymphocyte we have B cells, T cells, and all this role already we have discussed so far. So if you can read that content, see cells of immune system, you can understand this B cell and T cell. Same thing what we discussed, same thing will be there. Okay, so that is about the B cells and T cells. I hope you understand about the lymphocyte. So lymphocyte nothing but B cell, T cell together. B cell and T cell both are produced in the bone marrow. But B cell matured in the bone marrow, T cell matured in the thymus. That is the difference between this B cell and T cells. B cell fights with antibody and T cells fight with the help of the cells like cytotoxic killer cells. That is the NK cells, natural killer cells. Whereas the myeloid progenitor cells, this myeloid progenitor cell has the potentiality of producing some cells, like it has some platelets. It can produce platelets. Platelets can be produced, and then as well as it can produce some RBC, that is the erythrocyte. RBC can be produced, and even some mast cells. The tissue is made up of cells, mast cells, and it also produces one more cell type here. This is the entire called myeloid. Then one more cell is called myeloblast cells. Blast means it is producing more amount of cells, like it produces another type of WBC. One type of WBC is lymphocyte. Other type of WBC we have got this basophil. Diagram part is there in the book, you can see it afterwards. Basophils are there. Neutrophils are there, neutrophils are there. Then we have got some eosinophils, and we got monocyte, and this monocyte only will convert into macrophages. This is a very important public 1 mark question. Okay, after infection, monocyte will get converted into macrophages. Don't forget it. Okay, then it is also having one more cell called dendritic cells. That cell separately I will talk. Okay, so all these are the cells which is produced from one bone marrow. That is why it is called pluripotent hematopoietic. Everything is related to blood cells. So pluripotent hematopoietic stem cells. Stem cells means one cell has the potential to giving rise to another type of cells, same type of cells. So here we got myeloid progenitor cell, lymphoid progenitor cell, then myeloblast cells. Okay, so all these are important myeloblast cells. Now here we got platelets are there, RBC, mast cell, basophil, eosinophil, neutrophil, monocyte, macrophages, plasma, B cell, T cell, all together lymphocyte. So everything including dendritic cells, all these are the cells of immune system. Is that clear? So the diagram part and all, it is there in the book. You can verify it afterwards. During the revision time also, we can see the diagram part, the basophil, eosinophil and all. Inside the diagram, the nucleus part you have to draw and explain it. Already in 11th standard we have discussed, but still when revision comes we will take it up. So one more content I will just deal with it and then stop it. So I'll go with a cell called Dendritic cells. An important 2-mark question. That is why separately I am just doing it, right? So the cell is called Dendritic cells, dendrites, or it is otherwise called as dendritic cells, or otherwise called as dendrites. Okay. Now, why it is called dendritic cells is, if you can see the diagram of it, it is like this. This is the structure of the dendritic cells. It will look like the first dendrons of the neuron. If you see the neuron, it will be the neuron's top first part will be the dendrites. So it is appearance or resembling the dendrites of the nerve cells. That is why it is called that dendritic cells. And it is covered with long and thin membrane. It is always covered. It is covered with long as well as thin membrane. Covered with long and thin membrane. Why the name? Because it is having the name of the nerve cells. And these cells also will be presenting. So this cell is called Apc cells. What is Apc cells? Antigen presenting cells, which will capture the antigen as well as present the antigen to the T helper cells. When you talk about T helper cell, the T helper cell is involved in 2 types of immunity, in CMI and AMI. Why CMI? CMI only it is getting activated. In AMI again it is activating B cells. So in the CMI it is activated by macrophages. In AMI it is activating the B cells. So this T helper cell is an intermediate between the CMI and AMI. Don't forget it. The T helper cell is for both cell mediated immunity and as well as antibody mediated immunity. It is for CMI and AMI. Don't forget it. Very important 1-mark question. Okay. And this type of cells, dendritic cells, are present on 4 different areas types. So the one is called the Langerhans. Okay, islets of Langerhans. And the second one, it is the interstitial cells. Interstitial cells in between. Okay, interstitial means it is in between the 2 cells, and it is there in the myeloid cells. On different areas you will have it. So we will talk this when we discuss about the 1 marks. Okay, and finally it is there in the lymphoid cells. Clear, man? Right. So that's all about this cells of immune system. Actually cells of immune system I have to teach one day ahead, only after a day only, but still you can understand this acquired immunity, that cells and all. I can just add it along with this. So next day, tomorrow, we will be discussing about the organs which is producing this B cells and T cells. Which organ? Bone marrow. And where it is getting matured? Thymus. So after getting matured, where it goes and settle down. So all these organs that we are going to study tomorrow. So we are going to study about the bone marrow, we are going to study about thymus, and we are going to study about those organs which these B cells and T cells are going to be settled down. Okay, so that's all today. So see you tomorrow. Thank you.